This procedure begins with preparing an array of ACP1 GFP by first growing a lawn of cells and then pin out the array on a fresh plate. At the same time, prepare a fresh copy of DMA. Next, we mate the ACP1 GFP array to the DMA and select diploids, which are then sporulated on sporulation media. 5 days later, the meiotic progenies are germinated on LRK media on which only MAT-A haploids will grow. Through 2 additional rounds of selection, we recover haploid cells carrying both ACP1 GFP and a gene knocked out on kanamycin. Finally, we can pick individual strains directly from the plate and observe the mitochondria morphology by confocal microscopy.
Hi, I am Shabnam Tabassoli from the Lone Lab from the Department of Cell and Developmental Biology at the Life Science Institute of the University of British Columbia.
Hi, I'm Jesse Chow, also from the Lone Lab.
Today we will show you a high-throughput method for incorporating organelle GFP markers in various mutant backgrounds in yeast using a robotic system.
We use this procedure in our laboratory to study the morphology of organelles such as the endoplasmic reticulum and mitochondria.
So let's get started.
For this protocol, we use 2 different yeast strains. ACP1 GFP is a strain with a C-terminal GFP tagging of ACP1, which is generated by a PCR-mediated homologous recombination using plasmid PKT128. The strain background is BY7043. We also use the DMA strain, or deletion mutant array, which is an array of about 5,000 deletion mutants of non-essential genes. All of these non-essential genes were knocked out on G418. The strain background of the array is BY4741. To begin, grow a lawn of ACP1 GFP cells by pouring a 5 ml of overnight ACP1 GFP culture onto a His plate. Allow the plate to dry sufficiently by a flame or in a biological fume hood. Once dry, incubate the plate at 30 degrees Celsius overnight. After incubation, use a robot to array the ACP1 GFP strain into 1,536 colonies per plate. At the same time, prepare a fresh copy of the DMA by replica pinning to YPD G418 plates. Afterwards, incubate the plates at 30 degrees Celsius overnight. After the strains have grown, mate the ACP1 GFP strain with the DMA by replica pinning and overlaying the colonies on YPD plates. Incubate the plates at 30 degrees Celsius overnight. After incubation, replica plate the colonies onto SD His G418 plates in order to select diploid cells. Incubate the plates at 30 degrees Celsius overnight. In order to begin sporulation and germination, first replica plate the diploids onto YPD G418 plates to increase the efficiency of sporulation in the next step. After incubating at 30 degrees Celsius for 1 day, replica plate the diploids onto enhanced sporulation media, which contains reduced levels of carbon and nitrogen sources to induce the formation of meiotic spores. Next, incubate the plates at 25 degrees Celsius for a minimum of 5 days. After incubation, germinate the MAT-A meiotic progenies by replica plating the colonies onto LRK media. This media will induce the germination of haploid cells from spores. Incubate the plates at 30 degrees Celsius for 2 days. In order to select cells carrying both the GFP allele and the single mutant allele, MAT-A cells are replica plated onto LRK G418 media, which selects for haploid cells that carry the gene deletion. Incubate these plates at 30 degrees Celsius overnight. Next, replica plate the MAD-A meiotic progenies onto LHRK G418 media to select for growth of single mutants that also harbors ACP1 GFP. Incubate overnight at 30 degrees Celsius. The plates can then be stored at 4 degrees Celsius for up to 3 months. In order to visualize the desired mutants expressing ACP1 GFP directly from plate, start by picking the desired colonies from the plates. Grow the cells at 30 degrees Celsius in YPD or SD His media to early log phase. Finally, visualize the desired mutants using confocal microscopy with the GFP filter set. In this confocal image, a mutant and a wild type with the ACP1 GFP marker were visualized by confocal microscopy to study the mitochondria morphology. This particular mutant exhibits disrupted mitochondrial phenotype and is therefore a good candidate for further investigation.
We have just shown you how to efficiently incorporate a mitochondrial marker, ACP1 GFP, into the deletion mutant array by using a robotic system.
When doing this procedure, it's important to make the media freshly and allow the plates to have sufficient time to dry. If they are too moist, condensation will build up, increasing the chance of contamination. Also, you should double-check that all selection media are made correctly because they are critical to this protocol.
This procedure can also be used for other types of markers. For example, to visualize ER, we routinely use the marker ERG11 GFP.
So that's it. Thanks for watching and good luck with your experiments.